E. coli is a bacteria found within our digestive tract, and it is necessary for our development and proper functioning. We depend on the bacteria to break down undigested food in the large intestine and convert it into K and B complex vitamins. E. coli can survive with or without the presence of oxygen, which makes it a useful model organism in the lab. For our experiment, we are incubating E. coli bacteria in glucose, fructose, and lactose for 4 days. Our hypothesis is that the bacteria prefers glucose sugars.
The materials we used was microliter pipettes, vials of water, vials of E. coli bacteria, vials of 15% of glucose, lactose, and fructose, capillary tubes, timer, blood agar plates, Bunsen burner, flint, spinner tray, metal loop, alcohol 75% ethanol, masking tape, Sharpie, test tube tray, and biohazard waste container.
We started by making a control. Begin with 5 milliliter tube of sterile water. With a microliter pipette, remove 15 microliters of the sterile water and label the tube number 1. With a different microliter pipette, remove 15 microliters from bacteria culture tube. Add the 15 microliters of bacteria into the tube number 1. Place capillary tube into a second tube of sterile water and fill capillary tube to the line. Place the capillary tube into water tube number 1 for 30 seconds in order to tempt the E. coli. Empty contents of the capillary tube into sterile water tube number 3. Deposit 15 microliters from tube 3 onto 3 separate plates. Sterilize the metal loop with Bunsen burner and evenly spread. For test plate, we repeated the procedure for the control, but instead of water, we tempted each sugar with all 3 sugars.
Our results for our sugars after 4 days of incubation were the number of colonies in each of the 3 plates for E. coli and fructose tempted by glucose was 3, 2, and 4. For E. coli and fructose tempted by lactose was 2, 7, and 2. For E. coli and fructose tempted by fructose was 1, 3, and 0. For E. coli and glucose tempted by glucose was 1, 0, and 1. The number of colonies in each of the 3 plates for E. coli and glucose tempted by lactose was 0, 0, and 1. For E. coli and glucose tempted by fructose was 4, 0, and 1. For E. coli and lactose tempted by lactose was 0, 1, and 0. The number of colonies in each of the 3 plates for E. coli and lactose tempted by glucose was 9, 9, and 38. For E. coli and lactose tempted by fructose was 4, 23, and 6. Our results for our control, which was E. coli and water tempted by water, were 4, 3, and 1.
Analysis of the results reveals that most plates yielded comparable or lower numbers of colonies than the control, meaning no preference for the tempting sugar was indicated. The only clear conclusion that can be drawn is that both glucose and fructose are preferred by E. coli over lactose. However, no evidence for glucose being preferred over fructose was found. Therefore, our hypothesis was not supported by the results of this experiment.